morning, Dr. Nana and my fellow friends. We are from Group Rise. Today, we will be presenting our topic, which is micropropagation of Zingiber officinale, also known as ginger. Ginger has been a staple in traditional medicine and cuisine across the world, owing its versatility to two primary components, gingerol, shoggols, which provide antibacterial, anti-inflammatory, and antioxidant benefits. Ginger normally grows in tropical regions such as Malaysia, Thailand, and India, and it's used in both culinary use and medicinal applications. While ginger is in high demand, its conventional propagation through rhizome faces several challenges. First one, low multiplication rate, as ginger rhizome take months to mature and can only produce a limited number of new plants per year. Second, soil-borne diseases. Diseases like rhizome rot severely impact yield and quality. Third one, labor-intensive process. Manual planting and harvesting requires significant human effort and resources. Last but not least, productivity decline. Over time, continuous cultivation leads to reduced soil fertility and diminishing yields. So, to overcome the challenges of conventional propagation, plant tissue culture is emerging as a groundbreaking solution. Plant tissue culture involves the sterile cultivation of plant tissues under controlled conditions. This method offers several advantages. The first one would be disease-free plants, as plant tissue culture produces genetically identical plants free from diseases, ensuring healthy and robust crops. Second, elimination of soil-borne illnesses. By propagating in sterile environments, it completely avoids the risk of soil-borne diseases. Next, rapid multiplication. High-quality plants with desired are generated significantly higher rates, making it ideal for large-scale farming. Last but not least, labor reduction. Plant tissue culture minimizes manual planting and harvesting, reducing labor costs and effort. So we actually aim to determine the effect of exposure time to Clorox in sterilizing ginger to get clean culture, and to determine the effect of different auxins on callus induction and regeneration. Before proceeding to the plant tissue culture, it is crucial to make sure that the plants are fully sterilized, so we review literature to plan out the proper sterilization procedure. For the methodology part, the buds were cut out with a scalpel and the outer skin was then removed. After rinsing and brushing off the buds with tap water, they were then immersed in fungicide for 30 minutes. Afterwards, the subsequent steps were conducted in a laminar airflow. In the laminar airflow, the buds were dipped in 70% of ethanol for 60 seconds. The buds was then immediately transferred into an autoclave jar bottle containing 17% Clorox solution with 2 drops of 20 and kept for 30 minutes. For the second protocol, the exposure time for the Clorox solution was changed to 50 minutes. During this period, the jar bottle was frequently swirled and using a shaker to ensure all the surfaces of the buds came into contact with the sterilant. After 30 minutes, the sterilant was decanted and the buds were washed 3 times thoroughly with fresh autoclave distilled water to remove all traces of sterilant. Notes: interchange the jar bottle while washing. Next, the buds were dried before placing on the media. The buds were then cut in half and placed in 240 media for plant tissue culture.
Okay, now I'll be talking about the result that we obtained from our plant tissue cultures. And here's a table that shows the observation of culture from first sterilization with different exposure time of Clorox. And from figure 1, the graph shows the dynamic of clean culture counts across 50 days. And there were 4 replicates we had prepared for each protocol, but at the end, there is only 12.5% of viable clean culture left by using protocol 1 and 25% of clean cultures from 15 minutes of cross immersion. And here is also 4 pictures that attach showing the significant observations from first sterilization. Figure 2 is the fungus that grow in our cultures, and figure 3 is the buds that observed on day 22, which is the clean cultures that ready for subculture. And figure 4 is the most vigorous clean culture that we had obtained from this sterilization. Figure 5 is the root growth after we subculture the buds.
Okay, now is the result for second sterilization. We had observed our cultures across 43 days, and figure 6 is the comparison of clean culture counts between protocol 1 and protocol 2. We have prepared 4 replicates for each protocol. But at the end, there's only 75% of viable clean culture left for Protocol 1 and 50% of viable clean culture for Protocol 2. We also had summarized some of the significant observations for second sterilization. Figure 7 shows the bacteria infected cultures that we didn't obtain in sterilization 1, and figure 8 is the subculture that infected with fungus, and figure 9 is the clean culture that we obtained.
Okay, here's the observation for third sterilization, and this time we had only observed our culture until day 21st. But something different is we had prepared 5 replicates for each protocols. At the end, there are more viable clean culture obtained from Protocol 2 comparing with Protocol 1. And here are the figures that showing some interesting observations. In figure 1 showing the buds that turn black before it develops the shoots. And figure 12 shows the buds that didn't grow at all. And figure 13 is the bud that grows in a short time. The summary for sterilization result is that the contamination rate is higher in Protocol 1, which is the 30 minutes of Clorox exposure. Other than that, we also observed that the larger buds will exhibit faster growth, and the buds that were small won't develop shoots even culture for many days. At the same time, the contamination rate against time is lower across first attempt to third attempt, indicating that our hands-on skill had improved over time. And some of the bud will turn black before it develops into the shoots. In conclude, 15 minutes of Clorox exposure time is more effective for sterilization. And lastly, the figures of 14 were some of the subcultures throughout the 3 attempts. Next is the observation of callus induction with using the leaf of ginger, and there are totals of 8 replicates we had done. But at the end, there are no any significant callus formation observed, even though there is still some changes observed from the cultures, such as the decolorization of the leaf, and also some leaf cuttings has shrunk. Besides, there is also no any contamination occurs.
For discussion, we compare clean and contaminated cultures. The bar graph shows that the Protocol 2 with shorter Clorox exposure achieved a higher clean culture count. This balanced the minimized tissue damage while effectively reducing the contamination. The use of 2,4-D in the media and oxy is known for the promoting callus formation. So on the right, you can see the contamination observed in some culture, likely due to the endogenous microorganism or inconsistent handling. This highlights the need for optimized sterilization processes. Our finding also emphasize the hormonal balance required in tissue culture. Cytokinin like BAP and TDX favors the shoot proliferation but inhibits the callus formation. In contrast, auxin-dominant media such as 2,4-D effectively induce callus by promoting de-differentiation. This demonstrates how media composition directly impact the tissue response. These findings are crucial for refining the plant tissue culture technique. Lastly, our study had limitation. A small sample size limits the generalization, and contamination from endogenous microorganisms highlights the need for improved pre-treatment step. Despite this, the study confirms that the importance of balancing auxin and cytokinase for tissue culture success, paving the way for more efficient propagation protocol.
In conclusion, we have successfully demonstrated that a shorter Clorox exposure time, which is 15 minutes, minimizes tissue damage while effectively sterilizes explants. 2,4-D media was able to promote callus induction. However, BAP and TTZ medias were able to inhibit callus formation but promote shoot proliferation. However, challenges such as endogenous contamination and small sample size limit the consistency of these findings. Further optimization and validation of sterilization protocols and media composition could enhance the efficiency of micropropagation for large-scale applications. And this is our reference. Thank you.